Hi there, it's just Duncan from Axol here. We're going to ADPD, the conference next week, and thought I'd just go through some of the slides that we're going to be sharing with clients and colleagues there to give you an idea of how we envisage building a patient stratification model to support Alzheimer's disease. ADPD, it's next week. Our CEO, Liam Taylor, will be there, and you can catch up with him and also Chris Ward from Stratistem. You can read this at your leisure, but why do we feel like there needs to be a new, different way to approach AD? Well, primarily because we know the systems that are being in use just now aren't giving the results that people want. There's a lot of people working really hard in AD. And when we look at the model systems and sadly the failure rate of clinical trials, there's a real push to do something different. And what does different mean? Well, perhaps greater diversity in the models. We know the kind of polygenic and complex nature of AD. And maybe from that complexity, we can get a better understanding of the underlying genetics, and then bring that back into an understanding of drug action and ultimately, who would you pick in a clinical trial setting? So if we can get a better understanding and embrace the complexity of the model, then perhaps we can design better clinical trials, fewer people, more of the correct people, and greater success. So we've been working in partnership with Stratistem to put together the foundations for a new style of model, which is based on iPSCs. What are iPSCs? So iPSCs are induced pluripotent stem cells. And the whole connection, what I think is really important about stem cells, it relates back to a patient, a person, putting their arm out and donating a blood sample. That blood sample is turned into a stem cell, and from those stem cells, you can manufacture a range of relevant functional cell types. So for AD, we might be talking about cortical neurons, inhibitory neurons, neuroinflammatory cells, and glial cells. So maybe mentioned in the first slides, but how best to model AD. There's lots of different ways, and there are all surrogate models. So what's wrong with a mouse? Well, mice famously don't get Alzheimer's. Which mouse would you design? Which genotype would you design? Equally, same argument would relate to individual cell types or individual cell lines. So what we and many others are trying to be thinking about is embracing complexity, getting multiple lines or multiple people turned into stem cells, turned into neurons. So ultimately you get probably a very expensive cell culture dish which has dozens of patients in it and in a co-culture model. And by embracing that in gender balance, age-relevant patients, you can build up a complex cell culture model system to really understand the diversity of AD. And so, as we recently announced, Axol, in partnership with Stratistem, have rolled out a new library of lines. So here are 20 lines, which have a range of APOE genotype status. And the Stratistem collection comes from patients who live around the Manchester area in England. And we have Stratistem have collected full consent and ethics, but also medical histories of these people. And what Axol can do is we have the primary materials turned into stem cells. We can manufacture relevant cell types. So we're ready to be building these systems. What would we do once we have these cell culture systems? So Axol's competency is in manufacturing, but also looking at the functional endpoints. And there's a whole slew of endpoints we can look at for drug discovery, but then kind of rolling into this kind of clinical trial setting. And when we build up these models, we already have a very good handle on monoculture systems, so individual cell types. So there's microglia. This is some data looking at TREM2 mutation in microglia, very relevant for Alzheimer's. And a phagocytotic acid. These are functional acids we can run in-house. So we've got a good understanding of what these cells might be looking in monoculture. And again, back from diversity, there's a whole world to explore about individual genetic effects and how they may present themselves as functional phenotypes. And again, we've got a lot of expertise in building from individual cell types into co-culture systems, that's mixtures of cell types. And here we are dropping in ApoE4 astrocytes into co-culture, for instance, and measuring endpoints. So what would this really look like in terms of a big model building exercise? Well, we see this as a cooperative model building where together with partners, we come together to try to build and establish and characterize a gold standard AD model. In practice, we need a number of people in this group. We need people to provide materials. So we have our partnership with Stratistem. We see ourselves as manufacturers and assay development. We need design inputs. We need people who say, yeah, if you built that, that would be really useful because we have these programs of work. So we have design inputs, then we have primary users, and together in a consortium effort, we would look to build and then validate and characterize and then get first use of these systems. And we have whole Gantt charts around how we would do this. None of this is massively quick or cheap, but it's actually quite achievable to get to that first scale. And being in this group, there'll be certain terms around being inside this consortium, but the important thing is once we have a good understanding of its utility, then we make it accessible for researchers and drug discovery companies to come in and use this, because we're all here to try and do some good in this space. So, as I said, my colleagues will be at ADPD. If you'd like to meet them there, I'm sure you can just reach out to maybe Liam on LinkedIn, or you can just contact Axol and put you in touch. We also have information around the Stratistem collection and some really interesting modelling work we've done with a company called Tessera. Okay, hope that was interesting, and if you want to learn more, drop me a note. Bye.